wait for these. Okay, so before we start, I'd like to acknowledge the traditional owners of the land in which this event is taking place, the land of the Wurundjeri people of the Kulin Nation, and pay respect to their elders and families, past, present and emerging. So my name is Jane Oliaro. I run a lab here at the Peter MacCallum Cancer Centre in the Immunotherapy Centre. And today I'm going to be the MC of our first edition of the VCCC Monday Lunchtime Live. So for those of you who are new, a brief explanation about the VCCC. So it stands for the Victorian Comprehensive Cancer Centre, and it's a powerful alliance of 10 successful Victorian organisations committed to cancer control. Its role is to coordinate the collaboration between its member organisations and to develop a strategic program of work that integrates research, education and clinical care to maximise patient outcomes in a cost-effective manner. So welcome to today's presentation. We have some very interesting speakers coming up in the program, which you can see above me in the slide, so please register your interest at the VCCC website. So today we welcome 4 VCCC affiliated researchers. And these researchers have recently deciphered a new way that immune cells detect infections and cancer. Today you'll hear from 4 of our speakers, Professor Jonathan Sabon, Immediate and Past Medical Director of Cancer Services at both the Olivia Newton-John Cancer Wellness and Research Centre at Austin Health and the Olivia Newton-John Cancer Research Institute. He's also Professor of Medicine at the University of Melbourne and the School of Cancer Medicine. We'll also hear from Dr. Andreas Behren, who is also from the Olivia Newton-John Research Institute, where he heads a lab investigating tumour immunology. And we also have 2 speakers from the Doherty Institute. We have Professor Dale Godfrey, who is a laboratory head and the immunology theme leader at the Doherty, and Dr. Adam Aldrich, Senior Research Fellow from the University of Melbourne and Doherty Institute. So today's presentation will run through how this team have discovered a new molecular target for gamma delta T cells, known as B2N2A1. This molecule is present on multiple cancer types and is directly recognised by a type of immune cell called a gamma delta T cell. They've published their work in Science magazine, and their findings highlight a new type of therapeutic target that should engage the potent anti-tumour activity that gamma delta T cells are known to possess. So if you'll please welcome us in joining our presenters, and we'll first call on Dale Godfrey.
Okay, thanks everyone for coming along. So I'm the first in a 4-person tag team presentation. We have all 4 of us here because this is the result of a really great collaboration, and I think it's interesting to have each of our perspectives on how we got to this point and why we think this is an exciting development in the field of immunology and also for potential for immunotherapy. So I run a lab across the road at the Doherty Institute here. If you look out the door when you're heading out, you'll see the Doherty Institute. And my lab's interest is in what we call unconventional T cells. So T cells are a major part of the immune system, and there's all kinds of different T cells, and many types of T cells most people don't pay much attention to. These are the ones we call unconventional. And I'll spend the next 3 or 4 slides explaining this a little bit more so that you understand what we're talking about when we're talking about gamma delta T cells. So the immune system works by recognizing molecules that are either foreign or abnormal, and these molecules are known as antigens. And antigens can come from viruses or bacteria, but also some cancers, or most cancers and tumour cells, have their own particular molecules or antigens that can be detected by the immune system. So the types of antigens that most people in the field of immunology have been focused on are peptide antigens. So peptides that are derived from proteins that come from bacteria or viruses or cancers. And so these peptide antigens, and I've shown a couple of examples here with the pink and the yellow shapes. These are representing peptides from viruses or bacteria and tumours. These get taken up by specialised cells in our body called antigen-presenting cells, And these antigen-presenting cells take the peptides and they display them on the surface of these cells in the context of antigen-presenting molecules. And you can see here MHC1, MHC2. These are antigen-presenting molecules that are very good at presenting peptides to the immune system, to T cells that are passing by. So what we have in our immune system are these T cells. These are a major part of the lymphocytes in the immune system, and these are key directors of immune responses. Now, the T cells that most people think of when they're thinking about immune responses are these peptide-reactive alpha-beta T cells. And they're called alpha-beta T cells because they use T cell receptor alpha and T cell receptor beta genes. These genes are used to make proteins that form the antigen receptors that give the T cells their ability to see antigens. So these alpha-beta T cell receptors can interact with the peptide presented by the MHC molecule that is on the antigen-presenting cell. So this is how our T cells can tell that there's a foreign or a tumor-derived antigen that is present. Now, in addition to peptides, there are also other kinds of antigens, and these are less well understood, but there are antigens that are formed out of lipids, and so different bacteria have lipid antigens, and tumors have their own particular kinds of lipid antigens that can also be picked up by antigen-presenting cells and presented on the surface in the context of a different type of surface molecule called CD1. And this is also a lipid CD1 complex that's on the surface of antigen-presenting cells that can be seen by a different kind of T cell called NKT cells. And so NKT cells are also alpha-beta T cells, but they can see lipid antigens rather than peptide antigens. Then there's another kind of antigen called metabolites, broadly speaking. And these metabolites can come from bacteria and also, again, from tumors. And these can be picked up by antigen-presenting cells and displayed on the surface of antigen-presenting cells by yet another antigen-presenting molecule, in this case MR1. And MR1 plus metabolite antigen is detected by yet another kind of T cell, which are called mate cells, mucosal associated invariant T cells. And these are also alpha-beta T cells. They have an alpha-beta T cell receptor, but it's specialized for its ability to recognize metabolites presented by MR1. And then there's this fourth type of T cell, which we call gamma delta T cells, which is what the rest of today's talk is going to be focused on. These are T cells. They have got T cell receptors, but the T cell receptors come from a completely different family of genes encoded by the TCR gamma and TCR delta gene locus. And these T cell receptors are quite different from alpha beta T cell receptors. They see antigens in different ways, and it's not really known what sorts of antigens these gamma delta T cells can interact with. They can see molecules some of them see molecules like CD1 or MR1, and a lot of them see molecules of a family called butyrophilins. And butyrophilins are what we're going to be talking about, and the subsequent speakers will be telling you a bit more about. But there's really not very much that's been understood about how gamma delta T cells can see their targets. Why should we care about these cells? I think they're actually quite abundant. So NKT cells are up to 5% of blood T cells, mate cells are up to 5% of blood T cells, and gamma delta T cells are up to 5% of blood T cells. In the liver, for example, the mate cells are about 40% of liver T cells. So this is a very large abundance of these one type of T cell. And gamma delta T cells are dramatically increase in abundance in association with infections and cancer. So unconventional T cells, which are these last three types here, are a major part of the human immune system, and yet their therapeutic potential is very much underexplored, and that's what we were hoping to try and get to the bottom of. So gamma delta T cells, there's various different types of gamma delta T cells, and the types are based on which TCR delta genes they use. So we've got genes encoding TRDV1 or V delta 1, TRDV2 or TRDV3, V delta 2 and V delta 3. And these make different kinds of gamma delta T cells that can see different types of molecular targets. V delta 1 T cells can see things like CD1 family members. V delta 3 gamma delta T cells have unknown targets. People don't really know what they see. And then there's the V delta 2 gamma delta T cells. These cells see a type of antigen called phosphoantigens. And phosphoantigens are abundant in various microbial organisms, bacteria, but they tend to be more abundant in cancers. And so V delta 2 gamma delta T cells are able to see cancers, but the way that they can see these phosphoantigens and detect them is very much unknown. There was one molecule that's known to be important for this detection called butyrophilin 3A1, but nobody really understood how these gamma delta T cells were seeing butyrophilin 3A1 or phosphoantigen. Nobody was able to show a direct interaction between these compartments. This is what the following talks will tell you about. We've been able to work out how they can do this. So this is the focus of the rest of this series of talks. And just one last point, why should we care about gamma delta T cells in cancer? And there's a lot of studies that have shown gamma delta T cells can play a role in fighting cancer and killing tumours. But this one's quite an impressive demonstration. This is a meta-analysis of 18,000 different types of human cancer, samples across 39 different types of malignancies, where they looked at the different kinds of immune cells that were infiltrating the tumours and asked which immune cells are related to a favourable outcome and which ones are related to an adverse outcome. And right here at the top of the list, right here at the top of the list, we have the gamma delta T cells. And it's these gamma delta T cells that we need to understand. What are they doing in the tumours? Why does this relate to a favourable outcome? And so that's what the rest of this presentation will be about. And I've lost all control of the slide advancing. I don't know why. Sorry. Oh, it seems to be back. Here we go. And so the last point I want to make is that this work came about because of the Melbourne Immunotherapy Network. This is an organisation, I was the founding president, and it was designed to bring together immunologists and clinical researchers dealing with immunotherapy from all of these different partner organisations, because we know that some people are dealing with fundamental immunology and other people are dealing with immunotherapy, and these two groups of people don't get together and talk enough. And we figured that if we can have meetings where these organisations and these different scientists and immunotherapists can get together, we should be able to enrich both fields. And that's exactly what led to the discoveries that the rest of the speakers will tell you about today. And I think Jonathan will tell you a bit more about this at the end of the talk. So with that, I'll hand it over to our next speaker, Andreas Behren. Thank you very much.
Yeah, thanks, Dale, and thanks everyone for having us here. So I think what I'm going to talk about is a little bit really the history of that project, and I think that makes exactly the point that Dale just stressed, really, that you need the right forum to get the right people together to find answers you might not be able to find otherwise. This was a project that really started back in 2010, and it was really a pilot project which was a little bit like fishing for targets, and that's me sitting here like Homer Simpson, not exactly knowing what I'm doing, but trying to discover new targets on melanoma cells. That was with a collaboration with CSL Limited. And I'll take you a little bit through the different steps from target discovery to the results we had, and really when we were at a point where we had no idea what to do next. And I give you a little bit an introduction about the main method that we used here. So the method is called phage display, and the technical details are really not so important for how this method works. But what it really does is it gives you the possibility to really utilize genes that are encoding every possible human antibody. So antibodies, which are these molecules from the immune system, can really recognize every protein, peptide, whatever you can imagine, because they are highly diverse. And you can use that by using these genes and cloning them into something called phages, which are really viruses that infect bacteria. And then every of these different phages displays an antigen binding region of an antibody. And overall, you get a diversity of around 10 to the 10 billion different binders, so different antibody sequences encoded in these phages. And what you can do then, you have this phage library and you mix it with any target you are interested in. And then if these phages have a sequence that can recognize any of the targets displaced on the surface, it will bind, and you can elute it, grow it, and then sequence it to finally find out what it was binding. So we really used that to discover new targets in melanoma. And we did that because really Jonathan is someone who knows the value of clinical samples. So how we started is, that was a patient that Jonathan saw in the clinic at the Austin, and the patient had the accumulation of a lot of fluid. It was a melanoma patient, a lot of fluid, including a melanoma cells accumulated in the body of this patient, and it was drained in clinic, and Jonathan gave it to us in the lab. And what we did is we isolated all the tumor cells out of this liquid by using a specific surface marker on these melanoma tumor cells. And then we used flow cytometry, so we separated these melanoma cells, you can see them up here, from all the other cells that were within the sample. And that gave us many, many cells, and then we were able to use these freshly isolated tumor cells to do this phage display campaign. So we had these cells, we then added all of these different phages on top, and then some of them could bind surface molecules that were present on these tumor cells. And we then eluted those and repeated that a couple of times until we really found some of these molecules that were binding very strong to cancer cells. And we had some other control experiments to show that they are not binding to a large variety of normal tissues. The next step, what you do then, so here that gives you a little bit an idea about numbers. So we identified 2,200 different phages, or we could call them antibody-like molecules. And we knew that from these 800 were unique. So we're recognizing different things, but we didn't know what they were recognizing. We then repeated these screenings a couple of times to really enrich for the best hits. We had then 28 left in the end, and from these 28, we did a lot of control experiments to see what they are binding, how good they are binding, and then we were left with 6 from these 2,200 that we took further into additional experiments. So what we did now, we had these 6 antibodies or 6 phages, and we still didn't know what they are binding. We knew they are binding melanoma cells, but that's about it. So again, we put them together with our melanoma cells. We broke down the melanoma cells, we lysed them, and then we had a look if we now centrifuge out these phages and whatever they are binding to, can we identify with mass spec or Western blot what these proteins are, these targets that are bound by these phages. And that's what you can see here. So that is indeed the original first Western blot we ran with some of these 6 antibodies or 6 phages. And here you can see a strong band. So clearly some protein was pulled down here and recognized by one of these phages. And then we did a mass spec analysis to find out what is bound. So what was expressed on this melanoma cells that was recognized by these phages. And Dale already introduced this family of molecules already a little bit. And what came up is, so this hit was BTN2A1, one of these family members that was strongly expressed on melanoma and was identified from this one phage or antibody. And that was obviously very interesting for us and also interesting for CSL in that case, because there were some data emerging that were saying that these molecules are involved in immune regulation. And there were some hints that they were involved in immune regulation in also conventional T cells, mainly based on mouse models and different family members. So that was really, really interesting for us. And we thought, okay, we'll have a little bit of look into that. First step was we had a look, how is this molecule really expressed in different cancers and tissues, just to confirm our findings. And that's what you can see here. So that's gene expression based on nanostring. So really what it shows is in melanoma, every dot is a single melanoma sample, a fresh tumor. And you see a lot of expression of BTN2A1 in all of these melanoma tissues. And this is just a comparison to show you one of the other very famous immune regulatory molecules and their expression level within cancer, so PD-L1 and PD-L2. And we did not only see that in melanoma, we saw that in different cancers as well. And I think Jonathan will later give you a brief glimpse of some of the data for that. This one is now public available data from BioGPS, and this one is showing a little bit expression in a variety of normal human healthy tissues, and you can see there is not a lot of expression, but there seems to be expression in a lot of immune cells, but most other tissues seem to be clean. Obviously, that's important if you wanted to use something like that as a target at any time point. That's now a picture staining a melanoma specimen with antibody specific for that target, here shown in yellow, and you can see, yes, indeed, we can see this protein being expressed on the outside of all of these cells very highly, and if we verify protein expression again in different melanomas, each of these dots is one melanoma sample, and then counting the protein positive cells, percentage of protein positive cells, you can see it is widely expressed in melanomas, not in all cells, but in many, many cells. Next question, does it have a function in tumor regenesis? Because it's expressed in these tumors. So what does it do? We used some gene knockout experiments where we used melanoma cells, and we really got rid of expression of this gene for BTN2A1 to see if that has an effect. And here you can really just see how we generated these cell lines. So we transfected cell lines. You can see the cells that took up the DNA we inserted here in yellow, and that should really lead to knockdown or non-expression of this BTN2A1 in the cells that we transfected. And you can see that here, cells here would be the cells that were transfected where we wanted to knock out BTN2A1. And the histogram here, you can see, is shifted further to the left than in the other cells where we don't get rid of this gene. And that means that gene is not expressed anymore or lower expressed. And this is just a confirmation when we used here antibodies against BTN2A1. And you can see in wild-type cells, in non-knockout cells, we see these histograms here in orange and blue shifted to the right, which means they are highly positive for this protein, while the cell lines we created, the knockout cell lines, don't show this expression anymore. So that was really a generation of some tools. And then we had a look, does it influence the growth of the cancer cells? And you can see it doesn't, so it doesn't have a function here. We can also confirm that indeed this gene knockout worked on a protein level again, here in red or green, BTN2A1 stained in our melanoma cell lines. And here you can see these colors are gone. So clearly we got rid of our protein in our cells. The next thing, and I think Adam will also talk a little bit about those data, is that's nice. So we have a gene knockout tool now, but we were interested in, can we get an antibody that is targeting this protein? So something you could give in a mouse or in vitro or in a human that is binding to this protein and potentially inhibiting its function, whatever that might be. And together with CSL, just very briefly, you can see here all of these numbers. Is one antibody that we produced to bind this BTN2A1 molecule, and you can see we generated a whole library of antibodies. And if you see a color red, it means they are binding. Blue means they are not binding. And we had a look if they are binding specifically to A1 or other family members as well. And most of these antibodies are pretty good, and Adam will show some data generated with these antibodies. But again, using these antibodies and incubating melanoma cells with them, there is no change, at least not in proliferation of these melanoma cells. So no functional effect of these antibodies that we could see. We did a lot of other functional assays as well to get an idea what's happening. I told you people thought this molecule is an immune regulator, so we had a look in conventional T cell, alpha beta T cells, and if the antibody is influencing any immune reactivity or immune recognition by this arm of the immune system of the cancer. And unfortunately, we put a lot of effort into it. Nothing really worked. So we really had to sit down and think again. And as they mentioned, there is a couple of publications out there, mainly for people that are specialized in the field of gamma delta T cells, that would say these molecules are really related to gamma delta T cells, potentially this family of molecules. So we really had no idea about gamma delta T cells, and I'm sure that many other cancer immunologists don't know a lot about gamma delta T cells. So we decided we need some help because we don't know what to do. And then Jonathan used really the Melbourne Immunotherapy Network to present some of the data we generated. And that was really how we met Dale and Adam then. And I don't know if that's Dale or Adam, but obviously someone who was a little bit more clever than Homer, or us in this case, really then helped us figuring out what the whole story is. And I think Adam will tell us that. Okay, so I suppose while Andreas was doing all that work at the ONJ across at the Austin, Dale and I were at the Doherty Institute across the road, were getting stuck into gamma delta T cells and trying to have a look at answering some of the questions about their basic biology, I suppose. So this slide Dale already showed, but I've just flicked it up again because the subset of gamma delta T cells that I'll go into in more detail is this one in the red box. And as Dale mentioned, they recognize an unusual antigen or class of antigens called phosphoantigens. And they do this with their gamma delta T cell receptor. So it's the T cell receptor itself that is responsible for recognizing the phosphoantigens. However, there were a lot of unanswered questions regarding the molecular mechanisms for how this occurs. It was shown quite early in the piece that MHC molecules, which are important for presentation of peptide antigens, are completely dispensable for recognition of phosphoantigens. So it was something else that was important. And perhaps one of the reasons maybe why the knowledge of this subset has lagged behind a bit is because they're actually only exclusively found in primates. So rodents or other animals don't seem to have this population of cells, and as such, studying the complex interplay between these cells and the other cells of the immune system is quite difficult. Nonetheless, they are quite abundant. So as Dale said, they actually represent, on par with mate cells, the most abundant antigen-specific population of immune cells in healthy people. So about 5% of circulating PBMCs fall into this category of gamma delta T cells. And so in terms of what they recognize, these phosphoantigens are actually, so they're non-protein molecules, and they are intermediates in the synthesis of another family of molecules called isoprenoids, which are actually a large family of organic molecules, over 30,000 described, that are vital for all cellular life forms, essentially. So all bacteria and animals and plants use these isoprenoids for synthesis of other molecules such as steroids, cholesterol, vitamins, basically you name it. And in fact, off topic, that isoprenoids are one of the oldest identified biomolecules in evolution. So they've been identified in sediment that's over 2.5 billion years old. And it appears at some point in evolution, very early in the piece, so basically a long time ago, there were two different pathways for the generation of isoprenoids that appeared. Most bacteria use a pathway called the non-mevalonate pathway, whereas animals and many other eukaryotes use another pathway called the mevalonate pathway. And although the end products of these two pathways are identical, the intermediates in their synthesis are distinct. And gamma delta T cells, the phosphoantigen reactive subset, have evolved to recognize the bacterial forms of phosphoantigen with really high affinity. So they have a stimulatory potential of less than 1 nanomolar. So they're, to my knowledge, the most potent natural antigen that's been described. And that being said, even though the bacterial forms are really high affinity, the gamma delta T cells can still recognize the mammalian forms of phosphoantigen as well, such as this isopentyl pyrophosphate. But they're about 1,000-fold lower affinity, and so
in cases where there's transformation, such as in many tumours, you get an overexpression of these molecules, and the gamma delta T cells can get activated and kill the tumour cells in response. So why do we care about this particular subset? I'll just quickly outline two reasons. The first one is because they do appear to be important in immune responses. And this is just, you don't need to look closely at this table, but basically it shows that in virtually all bacterial infections, you get an expansion of the phosphoantigen reactive gamma delta T cells in people, indicative of an immune response. And as part of that, they proliferate, they produce cytokines, in particular interferon gamma and other pro-inflammatory cytokines. They exert cytotoxicity, and they activate other cells of the immune system. And you can, in terms of in vivo evidence, direct in vivo evidence of their importance, if you transfer PBMCs, so that's blood cells from healthy donors, into mice, and you challenge the mice with a bacteria, the mice can clear the bacteria or largely survive it. However, if you deplete the blood cells of the phosphoantigen reactive gamma delta T cells beforehand, and you only inject those remaining PBMCs into the mice, they largely succumb to the infection. So that indicates that the gamma delta T cells are indeed important. But perhaps more relevant to this forum is their reported role in cancer immunity. And so just briefly, it's been shown that this subset can kill a wide range of laboratory tumor cell lines in vitro, and especially when they're stimulated with phosphoantigens beforehand. And they're also associated with positive outcomes in patient cohorts who have had ALL and other similar diseases, where the more gamma deltas you have, the more higher chance of survival that you have. And as a sort of result of this evidence, there have been a number of clinical trials, so at least 15, just summarised here, to try and harness the immunotherapeutic potential of the gamma delta T cells in the clinic. And there have been some encouraging results, but by and large, by virtue of the fact that we still don't really understand how these cells work, it's difficult to properly harness their immunotherapeutic potential. So that, I suppose, that's leading towards our hypothesis that we were working on. But just before I go into that, I'll give a brief timeline of the key findings that were made on this particular population. So back in the early 70s, there was the landmark discovery of the alpha beta T cells, that they recognized peptide antigens in the context of MHC, and that was obviously the Nobel Prize-winning discovery of Peter Doherty and Zinkernagel. But gamma delta T cells weren't discovered until quite a bit after that. So in the mid-80s, they were discovered as a population, and it wasn't until the mid-90s that phosphoantigens were identified as the antigen for this subset. And in the years subsequent to that, it was found that these phosphoantigens needed cell-cell presentation, so they couldn't just be activate gamma deltas in a soluble form. But the biggest breakthrough came in 2012 when it was found that a butyrophilin molecule called BTN3A1 was essential for the recognition of phosphoantigens. However, it was unclear how it was working, because no one could really show that it was actually directly binding to the gamma delta T cell receptor on the adjacent gamma delta T cell. So the state of play as of recently was that the mode of phosphoantigen presentation was unclear. And there were various models that were presented to try and explain that, which I won't go into detail, but it was certainly an open question for a very long time. So while Andreas was fishing away for melanoma targets, we had our own little fishing expedition, albeit with a different approach. And what we decided to do was to make a soluble version of the T cell receptor to use as a probe for ligand discovery. And we did this by creating a tetramer. So that means we created a macromolecular complex of 4 different T cell receptors conjugated to a fluorescent molecule. And we used that reagent to stain a panel of laboratory tumor cell lines that were known to be, many were known to be targets of gamma delta T cell activation and killing. And indeed, we found that we had a panel of different gamma delta TCR tetramers that were all had slightly different sequences. But we found in particular that it was a melanoma cell line that Andreas was working on, the LMMel62 cells, that really lit up like a torch. It bound really strongly to these gamma delta TCR tetramers, suggesting that these tumor cells were actually expressing a ligand for the gamma delta T cell receptor. And we wanted to know what that was. And in order to try and figure that out, we utilized a whole genome CRISPR-Cas9 screen. And I'll just briefly describe what that is. So basically, you take you make a virus library that contains, in this case, the library that we used was about 120,000 different DNA fragments that are each capable of knocking out the function of a gene. So this is a designed library where every single gene in the human genome was targeted with 6 different guide RNAs. And you infect the target cell of interest with this library of viruses. And what happens is you get a heterogeneous population of cells. And if you do it right, on average, each of the cells has 1 gene knocked out at random. And then you take those heterogeneous cells and you select for the ones of interest. So in our case, we selected for the cells that had lost the ability to bind to the gamma delta T cell receptor. We purified them as a population, and then we sequenced the guide RNAs, the original guide RNAs that happened to infect those cells, and tabulated them by sequencing to determine whether there was an enrichment in a particular guide RNA in the population that we'd selected. And indeed, that was the case. So this is just the analysis of that screen. And there were only 2 statistically significant gene hits that we pulled out. The top hit was another butyrophilin family member, BTN2A1. And so this perked our interest because obviously we knew already from previous publications that BTN3A1 had already been implicated. They have similar names, but they are completely different proteins. So we next validated that finding by, and this is, I suppose, where we connected with Andreas and Jonathan, who were already working on BTN2A1 for different reasons, and already had the tools established, such as the antibodies and the knockout cell lines, and it was a really great situation that we found ourselves in. So we were able to quite rapidly take those cell lines that Andreas and Jonathan had made, and sure enough, when we stained these cells with the TCR tetramer, they don't, compared to the control cells, they've completely lost the ability to bind to the tetramer. And if we put BTN2A1 back into these cells, it restores the ability of these cells to bind to the TCR tetramer. And that correlates with the level of BTN2A1 expression on these same cells. So this suggests that indeed BTN2A1 was critical for the binding of the gamma delta T cell receptor tetramer reagent that we'd made. So we were able to then validate that the BTN2A1 was indeed a ligand for the gamma delta T cell receptor. We did that in a couple of ways. Firstly, we made a tetramer out of BTN2A1 itself. So that's the reciprocal tetramer, I suppose. And we stained blood cells with that tetramer. And sure enough, it stained a little population of cells in the blood that were T cells. And it turns out were actually exclusively gamma delta T cells. And then we performed another molecular analysis using soluble protein called surface plasmon resonance which measures the binding between two soluble molecules in solution. And we found that indeed the BTN2A1 protein was able to bind with quite reasonable affinity. So this number is a measure of the strength of the binding between the two. Bound quite well for a T cell receptor compared to the control here where there was no binding. Obviously, the key question that we then needed to answer was whether this was a meaningful interaction with respect to gamma delta T cell biology. So we addressed this in a number of different ways. One of which is shown here. So in this assay, we take blood cells from healthy donors and we culture them in vitro for 24 hours, either alone, which is the little columns in the left, or when we add a phosphoantigen stimulating compound called zoledronate. And so that activates the V delta 2 positive phosphoantigen reactive gamma delta T cells specifically. And you can measure that activation in a few different ways. For example, CD25 expression on the gamma delta T cells or interferon gamma production in the culture supernatant. If we added a control antibody to this culture, nothing happened as expected. However, when we took one of the anti-BTN2A1 antibodies that Andreas and Jonathan had produced, it completely prevented the gamma delta T cells from being able to respond to the phosphoantigen. So they were no longer getting activated by it, and there was virtually no interferon gamma being produced. And we're able to sort of show this in other ways using the BTN2A1 knockout cell lines as antigen-presenting cells, showing that they were unable to support gamma delta T cell activation. So this data showed quite clearly that this BTN2A1 molecule was actually a really important player in the response, the ability of gamma delta T cells to respond to phosphoantigen. And Andreas was able to also perform an assay to test whether this molecule was important for the gamma delta T cell mediated killing of tumor cells. And he did this by taking the melanoma cell line that we've been using as a model. And in normal, using the wild type cells, if you challenge them with the phosphoantigen and mix them with gamma delta T cells, you get killing. So that's this solid red line here. However, if you do the same thing with the BTN2A1 knockout MEL62 cells, you don't get any killing of these cells. So this suggests that indeed BTN2A1 is important for the gamma delta T cell mediated killing of this particular melanoma cell line. So then we started diving into trying to understand how BTN2A1 was facilitating the activation of gamma delta T cells. And it was known that mouse, so this is all work done on human cells, it's been previously established by others that mouse or other cell lines such as hamster cells are unable to present phosphoantigens to human gamma delta T cells. So it appeared that the mouse cells lacked something really critical that prevented them from being able to sort of do the job that human antigen-presenting cells could. So what we tried to do is a gain-of-function experiment where we added in different butyrophilins to the, in this case we used hamster cells, to see if we could restore or engender the capacity of those cells to present phosphoantigens to gamma delta T cells. And shown along the x-axis here are the different butyrophilin molecules that we put into these cells. And on the y-axis is the measure of the activation of the gamma delta T cells. So you can see that if you the controls, you get, if you add in the phosphoantigen stimulus, which is the blue, you get no response. Likewise, just BTN2A1 by itself was no good. We also included BTN3A1, which was no good. And there's another family member which has been implicated in the process called BTN3A2, which by itself or with 3A1 did not have any effect. However, when we co-transfected BTN2A1 and 3A1 into the antigen-presenting cells, we were we were able to enable those cells to present phosphoantigen to the gamma delta T cells. And perhaps when we added in 3A2 on top of that, it may have even enhanced it somewhat further. So this suggested that the missing pieces on the antigen-presenting cells that prevented them from being able to activate human cells were BTN2A1 and BTN3A1. Interestingly, when we repeated the experiment where we transfected separate populations of antigen-presenting cells with these molecules and mixed them together, there was no activation, indicating that perhaps these molecules needed to work together on the same cell. And I won't show the data, but we were able to show that these molecules are associated very closely to each other on the surface of the antigen-presenting cell and appear to be working in concert. And this is my last data slide. And we wanted to look at where BTN2A1 was binding on the T cell receptor. And so these funny-looking shapes that you can see here, I've lost the mouse. I'll just maybe use this. So this is a representation of the T cell receptor molecule, and the red-coloured regions are the areas that we found which were binding to the BTN2A1. So it binds to the T cell receptor on the side of the molecule. But curiously, we also found another region of the T cell receptor on the other side of the molecule. But while it didn't bind to BTN2A1, it was absolutely critical in recognizing and responding to phosphoantigens, suggesting that domain of the T cell receptor is actually binding to something else. And given that BTN2A1 and BTN3A1 are working together, we propose, or the evidence that I suppose our data suggests, that a model whereby BTN2A1 is binding to one side of the T cell receptor, and BTN3A1, which is associated with BTN2A1 on the surface of the antigen-presenting cell, is binding to the other side of the molecule. And phosphoantigen, which is the stimulus, comes in, binds to the BTN3A1 molecule, which has previously been shown, and that delivers a signal to the gamma delta T cells to become activated. And a similar process may be occurring in tumour cells, which themselves also, as Andreas pointed out, can express quite high levels of BTN2A1. So at this point, I think I'll hand over to Jonathan, and he can finish things up.
Thanks, Adam. So I'm going to try to now address the question of what does all this have to do with human cancer and cancer treatment, and to really draw together some fragments, little threads of information, and perhaps to highlight some paradoxes as well that the data that we've seen presents. And I'm going to pose this in the way of several questions, and I really only just addressed the first two of these, but we've heard a lot about phosphoantigens and about innate immune responses. And so the first question is, what's the role of these innate immune responses to phosphoantigens in cancer? And in particular, in cancer treated with a class of molecules called bisphosphonates, and I'll come to those shortly. In terms of cancer recognition and immune surveillance, that's the process whereby the body continually searches for abnormal cells, particularly cancer cells, and eliminates those. And the recognition that is now increasingly strongly recognized that the microbiota, particularly in the gut, but probably in other sites as well, has some sort of impact upon anti-cancer immunity. And then there are additional questions which we won't address today, such as can these cells, these gamma delta T cells, be harnessed as anti-cancer agents? Can they be manipulated in vivo, or can they be adoptively transferred as cancer therapies? Now, turning to bisphosphonates, the top graph shows some data which is more than 10 years old now, which shows the effect of a molecule called clodronate on breast cancer relapse in the adjuvant setting. So this is a relatively small trial in which about 300 women were treated with standard adjuvant breast cancer treatment and then received a molecule clodronate in addition. And you can see that the addition of clodronate improved the survival of these women by reducing the frequency of cancer relapses. Now, clodronate is a molecule that acts on bone, and the assumption has been all along that this molecule was acting by changing the bony microenvironment and reducing the risk of relapse by affecting the interaction between tumour and bone. And in fact, another study was done using another bisphosphonate, this time zoledronate, the molecule that Adam specifically mentioned in his talk, in which zoledronate was added to standard adjuvant therapy. And you can see in this subset analysis that there was also an impact from zoledronate. Now, zoledronate didn't have an effect overall, but only on a subset of women in this clinical trial. That was a subset of postmenopausal women. So the question was, is this an effect on the bony matrix and the way tumour is likely to interact with bone? And that was the assumption all along. And so another study was done using a molecule called denosumab. Denosumab, some of you may be familiar with, inhibits a protein called rank ligand. And rank ligand also has effects on bone remodeling and strengthens bone and is used to prevent fractures in people who've got weak bones or bony metastases. But it doesn't act through this mechanism that Adams described by increasing the availability of phosphoantigens. And it was really interesting to see in a publication that's just come out just a couple of weeks ago that, in fact, this alternative way of manipulating the bone microenvironment had no impact at all. Now, this in no way says that zoledronate is acting through this phosphoantigen mechanism, but it's highly suggestive that there's something quite different when you compare the impact on bone from zoledronate versus the impact on bone from denosumab, and raises the possibility that, in fact, there is an additional impact here, and that is through this phosphoantigen generation mechanism. Now, this is data which Andreas has already shown you, and I just want to use this to highlight the fact that we found BTN2A1 through a search which was aimed at trying to identify targets in cancer. So it was abundantly expressed in cancer, and you can see from the immunofluorescence that in a proportion of melanoma cells it can be expressed at very high density all over the place. And recapitulating the nanostring data which you've seen, you can see that it's an order of magnitude higher than PD-1 and PD-L1 and PD-L2. So these are other molecules that are known to regulate immune function, and it's sort of interesting to see this molecule associated with some aspect of immune function expressed at very high level on a tumour cell. But if the molecule was there and was helping the immune system kill the tumour cell, that would not provide the tumour cell with a survival advantage. So there's a paradox here. And the paradox is that if it's there for a reason, and if the cancer finds benefit in having this molecule there at high level, then is it protecting the cancer? In other words, is it doing the very opposite to what we might want it to be doing? So let's just look at the tissue distribution, and you've seen this data already. Primarily, BTN2A1 is expressed on cells of the immune system and at relatively low level on a variety of other cells. If you do look at the other cells that it is expressed at, and this is some data from something called the Phantom, the Riken Phantom 5 project, looking at RNA, you'll see that there is expression in a variety of tissues at relatively low level, and these tissues are largely neural tissues. So the cerebellum, actually probably among the highest levels are in the cerebellum, but also in a variety of other neural tissues, and then a smattering of other epithelial tissues as well, such as prostate and pancreas, but in general, not expressed at significant levels in non-immune tissues. But then if you look at cancer, and this is extending on the data that you've seen in melanoma, if you look at a variety of other cancer types, and here we've got liver cancer, here we've got breast cancer, here we've got prostate cancer, and here we've got pancreatic cancer, the levels of expression of this molecule are higher in many of these cancers than in the normal associated tissues, which were not malignant tissues. So again, raising this paradox that if this molecule is assisting immune function, how come it's present abundantly on cancer cells? So there's another interesting tissue which sometimes express molecules that play an immunoprotective role, and that's the placenta. The placenta in addition to providing oxygen and nutrients to the developing fetus, has to play a critical role in establishing a barrier between the immune system of the mother and the immune system of the developing fetus. And if you look at a molecule such as PD-L1, which we've met before, you can actually see that PD-L1 is expressed on the placental trophoblast cells at fairly high levels. And you can see that brown staining all over the pancreatic trophoblast. And PD-L1 is well recognized as a molecule that plays this important role in protecting tissues from immune cell activation. So the question then, and this is data from others, is what does BTN2A1 expression look like in pancreas? And you can see that the pattern of distribution on human pancreatic trophoblast is very much the same as PD-L1. So all of this gives rise to the impression that it may be playing a protective role, which of course is the very opposite of what we saw from Dale early on, that if you look at gene signatures in tumour tissue, that the most favourable prognosis is associated with high expression of a gene signature associated with gamma delta T cells. So that's shown in the first panel. You've seen this already. But in fact, there's another paper that was just produced in October of last year looking at triple negative breast cancer in which something similar was seen. And that is that there were gamma delta T cells in the breast cancer. And if they were there, they were associated with cytolytic activity, with gamma interferon production, and far better outcome in this small group of breast cancer patients. So a paradox. So can gamma delta T cells do both? Can they be both helpful and unhelpful? And this is a little bit of data, again, produced very recently, just at the end of last year, which showed that, in fact, there can be gamma delta T cells that have suppressive function. This is some work that was done in vitro in which gamma delta T cells were mixed with alpha beta T cells from the same individual and were cultured, and the effect of the gamma delta T cells on the alpha beta T cells was evaluated, looking in particular at the proliferation of those cells. And it was seen that the gamma delta T cells were capable of suppressing the alpha beta T cells, but they only did it under certain circumstances. There needed to be a very strong TCR signal coming through to the gamma delta T cell, and the cytokine milieu, the microenvironment in which the gamma T cells were operating, needed to be optimal in order to get this suppressive effect. And this was mediated by V delta 2 gamma delta T cells, the ones that we now recognize as being presented antigen to by BTN2A1. So this is a function very much like regulatory T cells, and typically regulatory T cells are associated with a molecule called FOXP3. But if you looked at FOXP3 expression in these gamma delta T cells, it was not elevated, and rather this inhibitory activity was associated with PD-L1 expression, and that PD-L1 expression could be blocked with an antibody, and if you blocked it with PD-L1 antibody, you're able to release those cells from those suppressive effects. So there's a yin and a yang. These gamma delta T cells under some circumstances can be suppressive, and under other circumstances appear to be able to kill cancer and be associated with a favourable prognosis. And that seems to be context dependent and may well have to do with the sort of microenvironment in the tumour and the nature of the antigens the tumour is presenting and the strength of the signals that those gamma delta T cells are receiving. So I'll finish it up here. These are just emerging threads of evidence. We don't have answers to any of these questions completely, but clearly there's a lot of interest now in trying to understand how these cells work, what determines outcomes in terms of the clinic, and whether or not biphosphonates in cancer are acting in part through this pathway, and the important role that BTN2A1 may potentially play in immune evasion. So ongoing work that will be done through our collaboration and no doubt through others will be to define the role of these immune responses, in particular these innate responses to phosphoantigens in cancer recognition and surveillance. Understanding how microbial phosphoantigens may potentially impact on anti-cancer immune responses, how we can harness these cells as anti-cancer effectors, and as a sort of a more personal conclusion, the critical importance that the Melbourne Immunology Network has played in bringing together the opportunities that this collaboration has provided. So it remains to just finish up here. I'm afraid we're running close to time, but I'd like to particularly acknowledge Mark Rigow, who's a PhD student working with Adam and Dale, who did a lot of the later work in this project. and a list of other people from the Doherty Institute listed below him there. Simone Ostruka and Catherine Woods at our institute, and our collaborators at CSL in particular, Con Panousis, Gino Vairo, and Eugene Merezkovsky, and of course the three previous speakers. Thank you very much.
Okay, thank you very much. So we have time for a few questions, and I think if we can ask our speakers to sit up here, we might just try and answer some from the audience, as we only have a few minutes. With a question over here. They'll bring you the microphone if you can ask through the microphone. Thank you.
Since this molecule is highly expressed on your melanoma cell, and since you also have from your biopanning really good antibodies that will bind to that, have you considered bypassing the gamma delta and instead of making a radio-labeled antibody or a cytotoxic-labeled antibody to directly target those cells?
Yep, well done.
Yes, it's certainly an approach that can be taken. We haven't put principal focus into that, but that's absolutely a strategy that can be considered. Of course, you need to think of what the off-target effects might potentially be on other tissues which express BTN2A1, and that's something which would need to be modelled up and evaluated as a potential consideration, especially around toxic effects of such a strategy.
Did you use skin mice or something to model that and test it out?
Yeah, the BTN2A1 is not expressed in non-human primates, so the off-target effects, you get a good result in the mice, but it might not be an accurate reflection of what might happen in humans.
I do have any more questions. So maybe on that note, is there a equivalent of these T cells in mice that you could test some of these theories?
No, that's one of the funny things about gamma delta T cells. They're very different, and their targets are quite different in mice, and that makes it hard for us to study them in models. But you can do xenogenetic transfers of the T cells and tumour cells and potentially study human cells in a mouse environment to carry out some of these studies. But it's not easy because these V delta 2 gamma deltas are not in mice, and BTN2A1 is not in mice.
Sure. And maybe because we rushed the last bit, you could maybe just elaborate a little bit for the audience on where you're taking this study next and how you see this then moving from the laboratory more into the clinic.
Well, I guess how we take it from the laboratory to the clinic is a good question, and we are nowhere near doing that. So at the moment, we are really trying to find out what's really happening. Is it a useful target? Can we manipulate it? And how useful will it be really to use in potential cancer therapy? So I think we still have a long way to go and a lot to learn. One is we are trying to develop a mouse model so that we really have the chance to do some in vivo studies and get an idea what's happening.
Okay, great. So we are a little bit short on time. Does anyone have any burning questions before we thank our guest speakers for today? And I believe I have a present for you all. Okay, so we'll just put our hands together and thank our speakers and for making it over.